Good afternoon to all of you. After listening to your interest in the morning, I decided to change the way of the presentation. This was supposed to be for 30 minutes. Definitely, if we continue, nobody will be there in the audience. So, I've been asked to cut down to 10 minutes. Even in the 10 minutes, I'm going to show you the PPTs only for 5 minutes. So 5 minutes, I'm just going to tell you what it contains. So most of you know that by using pan parak and ganja, many people are getting neurodegenerative diseases. And it is going to be million in another 20 years. So we have been looking what are the neurodegenerative diseases, and main things are Alzheimer's diseases, Parkinson's disease, Huntington disease, and so on. Of course, Alzheimer's disease is coming because of the plaque in the brain, and Parkinson is due to motor problem, and both the things they have only limited medicine. Then we thought, will there be any medicine from nature? So these are called phytochemicals. So that I've been working. The past 5 years I've been working on the marine natural products like seaweeds and so on. We identified some compounds and we did how the work on how these molecules bind with the targets like acetylcholinesterase, butyrylcholinesterase, beta-secretases and so on, about which you don't know anything. Okay. So I will tell you about our field. There is a field called X-ray crystallography, which has produced more than 40 Nobel Prize winners. So that is the only unique field which has produced so many Nobel Prize winners, and most of them are for the novel structures obtained using single crystal X-ray diffraction. If you want to understand the function of a molecule, or if you want to modulate the function of a molecule by binding with a small molecule that is known as, which is known as structure-based drug design, the knowledge of the three-dimensional structure is important. If you look into the database called protein database, we have more than 1.5 lakhs these structures deposited. And whenever a structure is doing a malfunction, you are supposed to attack the active site with a small molecule to control the function. So that is the trick. There are automated programs for doing that. That's known as docking. So in the audience, I have my friend, one Raghu, who was a former global vice president of a module called Schrodinger. So now he is the proprietor of one more thing, Molecular Sciences Corporation, which will be started in December. So I have been actively using this thing for more than 10 years. So that's what I'm going to present to you. So as I told you, every molecule has atoms. If the number of atoms are less than 100, we call it a small molecule. If you look at proteins or enzymes, they are made up of amino acids or peptides, which contain thousands of atoms. Those are called macromolecules. Every molecule does a function. Somebody told you about sickle cell anemia, no? There's only one mutation of amino acid, the function changes. Like so many cases are there. So to understand the remedy for the diseases, you must first of all know what is the three-dimensional structure of the protein related to that disease. So that you have to design molecules to bind. That is known as structure-based drug design. A knowledge of the three-dimensional structure is required. That is why crystallography plays important role. To ask you one question, nobody is going to answer. Who is the person Indian, who shares the Nobel Prize for solving the biggest molecule? Don't nod your head. Who is the person? Itiraj College, people are asking so many questions. No, I'm also from Chennai. I must ask, no? There's an Indian who solved the biggest structure. And he shared Nobel Prize with 3 people. No. Because it's not in the syllabus. That's all. When Vidya was doing MSc, I used to take 5 hours classes at this stage, quantum mechanics, classical mechanics. Suppose I do this in these colleges, present days, people will get TC and go away. Right? Because continuously 2 hours you are unable to listen to science because you are in between break dance. Huh? So that is the level. Because now everything is fast food, no? So that is why I'm not showing PPT at all. Fast, no? I'm also fast food person, no? So now you know there are molecules which are doing some function. If they do some malfunction, you must bind a small molecule at the active site to control the function, to modulate. Right? To whatever level, suppose you have 100 molecules to modulate, which is the best one? How to take? Suppose you have headache, no? So many tablets are there. Most of them contain paracetamol. Which is the best one you have to take? High dose or low dose? You don't get headache. Maybe after 15 minutes you will have it. It is always better to have low dose. Because once you take high dose, no, after that no high dose is available in the market. No, that's all gone. So that is called technically IC50. What should be the inhibitor concentration of the molecule, small molecule, which is going to bind with the molecule to reduce the activity by 50%. Okay? Then I have to answer the suspense also. The holy name is Venkatraman Ramakrishnan. The structure was ribosome structure. So he shared it with Stiles and Ada Yonath. Okay? Because he was an Indian, we don't want to give credential. So another thing is, October 8th, a famous scientist was born in India. He missed the Nobel Prize for his famous discovery about the structure in our body. Can you guess who is that? Very good. G.N. Ramachandran. Is there any connection between G.N. Ramachandran and a Bengaluru professor who was a Nobel laureate? Who was in Bengaluru, who was a Nobel laureate? Out of syllabus. Sir C.V. Raman. He was director of Indians of Science, no? G.N. Ramachandran was the student of Sir C.V. Raman. He found the triple helical structure of collagen or muscle fiber. For double helical structure, DNA, Nobel Prize was given. To whom? So many unanswered questions in your mind. What's none? No biotech person is here, all engineering? Yeah, you are the only person who is answering. I'm asking, all others are other discipline? Biotech. But this is not in the syllabus. Very good. At least next time you include the syllabus. Nobel laureates of India or Nobel laureates who missed prize. Okay. So now people will be wondering why this fellow is crying without showing any stuff. Okay. So this introduction I given, what are the neurodegenerative diseases, Alzheimer, Parkinson, ADPD, and recently we found that there are something called small proteins called neurotrophic factors. There are 7 neurotrophic factors. They bind with some receptors and they start the signaling pathways which leads to neuronal death, causing these diseases. So now, we thought, can you bind those amino acids which bind with the receptor so that you can block the binding of that with the receptor and stop the neuronal death? So that's what we have done. We have taken about 47 chemicals from the natural products. And this is the reason of the Alzheimer and ADPD, major public health problem. AD is the most common form of dementia, accounting for about 50 to 70% of all cases. Then it is due to beta amyloid formation. Then you can compare left-hand side healthy brain, right-hand side Alzheimer and Parkinson. Do you find any difference? Don't say no difference. Then eyesight is the problem. You got to go to Agarwal clinic, eye clinic. Is there any difference between left side and right side? Healthy and Alzheimer, Parkinson. What do you find difference? No difference. All are healthy. Hey, what is there? If you have good eyesight, you can answer, no? Left side, right side. No difference at all? Simple lunch only, no? If you have been given lunch which I serve in conference, that's all, you'll be sleeping. Huh? Karabat, that thing. What's that? Sundal, we got what? That's all. Channa, parota. With 3 dishes itself, we are sleeping. Huh? You come and attend my symposium. October 8th to 10th, Jayanagar symposium. Okay. Now, what are the various stages of the symptoms? If you have mild neurodegeneration, memory loss will be there, lingual problems, mood deviation, moderate, I given reason. Severe means motor impairment. You have to be bedridden only, no more walking. Then this is a list giving the people living with dementia around the world. Then this is about global deaths and prevalence for Alzheimer's disease and other dementias. This is about Parkinson's motor problem. Only 3 medicines are there. You got to change the doses, that's all. Not yet. So this gives the mechanism of this Parkinson. Then this is the earlier work I did. For Alzheimer's, these are the 2 protein targets. We got to try structure-based drug design. For Parkinson, this is the other target. This is the macromolecular structure I'm talking, proteins. 150,000 structures are deposited by crystallographers in a protein data bank. It is free to use, okay? Yeah, if you look at the center, there's a small molecule, right, with ball and stick. This controls the function, okay? This is for acetyl, then Alzheimer, butyrate. So, based on our work, we have found left-hand side so many compounds from marine seaweeds which acts against the targets in the second column to control diseases mentioned in the third column. So then we went to terrestrial compounds. From sea we jumped to ground. So we talked about the receptor called P75NTR, neurotrophin receptor. Then also we talk about TRK, tyrosine kinase family. These are receptors which bind these neurotrophins and which initiate the signaling, causing neuronal death. So you have to avoid this binding, right? So this is the mechanism. This is neurotrophin signaling, explaining. So these are the structures. These are small proteins, structures available for this neurotrophin, 7 neurotrophins, okay? So this shows you how the neurotrophin 3 binds with the receptor. See, at the center in the blue, how comfortably it binds, it has interactions. So signaling process starts. So what are the interacting residues? That's what we are interested in. Sometime after binding, left-hand side, we have 2 molecules. So dimerization occurs with tyrosine K binding. These are not stories. You must know this is a science which you are not knowing at all. If you have free time to go to Google or something, everything will be there. You have free time, but you don't Google this, no? Some interested Googles are there. Huh? That's the problem. So now, these are the compounds I have taken. Everything is natural bioactive compounds. I subjected them molecular docking studies. Whenever you do the docking, there are 2 parameters. One is known as score, another is energy. Based on that, we select the compounds. So these are the best compounds we selected. 1, 2, 3, 4, 5. Among them, there are 2 techniques. One is known as virtual screening. Because whenever a small molecule binds with the active site, it produces changes in the rotation angles, that are conformation angles, the concept introduced by G. and Ramachandran. So, if you want to take into the rotation of each bond, that will take a lot of time, 1 day like that, no? So, initially, even though we know that binding causes your conformation changes, we assume initially that receptor is rigid. And you give flexibility only to ligand. Okay? Then, from hundreds of compounds, you can filter to 10 compounds. Now you go to reality. You assume both receptor and ligands are flexible. That is known as IFD, induced fit docking. It takes 1.5 hours like that. So that's what we did. Okay. So you're asking if this thing goes like that, you will cross beyond 10 minutes. No. So this is known as Lick plot, showing the interaction of the small molecule ligand with the amino acids at the active site. So same stuff, one I'm explaining. So similar thing we did for all the 7 things. So since you are not interested, there is no meaning in showing you all. You want the end of the story, no? I know. Because teachers are not interested, first of all. They have timing, no. That's the problem. I never used to go to colleges, don't mistake me. That is the reason. Dance, time complete 11, 2, tea break, something, that's all. But people don't care whether you have. When before attending conference, after attending conference, you got to do a scanning of MRI of your brain, no? Mostly it will be down nowadays. So don't mistake me, that's the reality. So now we'll tell you the conclusion. So 7 neurotropic receptors binding I have taken. If you look at the natural compounds, best compounds, I have only 5. Rutin, piniculatin, salvianolic acid, naringin, and rosmarinic acid. So now we are thinking to have a formulation of these 5 and to give to patients. And we have a big project from Ayush. So we are planning for about 9 crores. So with Nimans, they have Parkinson mice. So it's equivalent to treating a Parkinson patient, no? So that's what I'm trying to do. So if you have listened to this, you could ask questions, but unfortunately, since you didn't listen, I don't expect any questions. I thank both Dr. Bhuvneshwari and my former PG student Vidya for all the way coming to Chennai for invitation. That's why I come all the way from Chennai, Bengaluru to Chennai, Delhi, Bengaluru. So thank you. So we don't understand, you think that there is a field called X-ray crystallography, which has produced more than 30 Nobel Prize winners. Our Indian scientist, Sir C.V. Raman, was one among them. His student, Professor G. N. Ramachandran, was the one who narrowly missed the Nobel Prize. Okay, thank you. So if you have any questions, you can answer. Huh? Which plant? Yeah. That is secret, no. No, we did it. We did it.
It's an extracted compound.
No, actually we are going to do now, because we don't want to believe reported values. These are all from literature. You go to literature, natural compound, Alzheimer, natural compound, Parkinson, so many papers are there. I collected. Then from here, I go for PubChem like that, what are compound release, or GCMS result. Again, everything Google only. Scientific way, I utilize Google.
It's a database. If you put this, you will get the plants.
I didn't give any plant, no name. It may be one among them. I didn't give them. So I thank you. One question I received. So you are luckiest person to ask the question, no? Let us see what is the question.
You ask like that only, no problem.
Hello. Sir, I just had one doubt. So here, I think neurodegenerative diseases were used, but in certain neuropsychiatric diseases like autism spectrum disorder, VDNF plays a role in the development and the subsequent management. So can these compounds even be used to target neuropsychiatric diseases?
Yeah, but nobody is working on that, madam. That's the problem. Actually, why I was very much interested here is, my own father-in-law, come brother-in-law, he was suffering by Parkinson for 10 years. That's why I was very much interested. But nobody is there to work, no. Because the moment we work for 1 year, 2 years, they want publication. It's not impossible. This is not one man's work, right? So animal test, the ethics committee is there, human trial is there, which we cannot do. Luckily, Padmanabhan did his PhD in our department. He's head of biophysics in Nimans. So he said, we can do. So he's one of the collaborators. So that is the way. Everything is there, because before allopathy came, people survived, no? Right? People were there in the globe with the diseases. So now, when I went to Ayush, you won't believe, such crores of rupees they are wasting. I should not waste. Any politician is there? Okay. Sorry about the word using. Crores are spent without any outcome in nature, or they want nature, no? How many papers publish in nature something? Even IIC doesn't publish nature regularly, publication. But moment you get it, how many, you show me the index, how many papers you publish in nature, PNA, something, without knowing. So that's the story, Indian story. In spite of that, India is doing science, good science. In NIRF, South Indian University has come to the top, no? NIRF. That's a good thing. Instead of the political suppression, we are coming up. So we have to thank our own scientists. And now there is a boom for AI, ML. So no more science. Basic science, I mean. Yeah, everything is part of money, dollars, no. Yeah, I worked with the Nobel laureate. I got to stay there in US. I worked for 4 years. But had I stayed there, I would not have produced 44 PhDs in India. And those guys are very loyal to me. Until, wait, then clap. They're very loyal to me. Until they get the green card, they used to communicate with me regularly. They've got recommendation, that's all. 40 people stopped communicating with me. Because they're citizens of US. Thank you. I hope it's a lively talk which you people wanted, no? Audience.